Hello and welcome to our webinar this afternoon. My name is John Cooper. My colleague here is Dr. Deepika Devner from Delta Dot. In just a moment, Deepika is going to be going through her presentation for us on using a single technology platform to analyse monoclonal antibodies. The presentation is going to take about 15 minutes or so, and then afterwards there'll be a question and answer session. You can ask questions whenever you like. Just write them in the dialog box there, and we'll face them afterwards. Also, if you want to ask questions that are not related directly to the presentation that Deepika gives, please feel free to ask those as well, and we'll answer as many as we can. Okay, Deepika, it's over to you in the presentation.
Hello and welcome to this webinar. Today I'll be speaking about the analysis of the monoclonal antibodies Infliximab and Rituximab using the HPCE 512 capillary electrophoresis platform. The monoclonal antibodies Rituximab and Infliximab are biologics administered for the treatment of arthritis, as well as a number of other inflammatory and autoimmune conditions. A 50 ml vial of Rituximab for infusion can cost in the region of £1,000. Once treatment has been started, it has to be given fairly frequently. For example, Infliximab is generally given at time points of 0, 2 and 6 weeks, and is followed by infusions every 8 weeks after that, so it's easy to see how the cost of treatment builds up. Current protocol dictates that once an infusion is reconstituted, it must be administered within 24 hours. Any drug left in the infusion is discarded. This leads to high levels of drug wastage and contributes to the high treatment costs. In this presentation, we show that the HPCE 512 can be used to determine fragmentation, stability, shelf life, and post-translational modifications on these monoclonal antibodies. This data can be used to determine whether a drug is safe to administer following reconstitution, which could prevent wastage and lead to significant cost savings. The first mode of electrophoresis that was used was capillary gel electrophoresis, or CGE. CGE is a method by which SDS-coated proteins move through the sieving matrix and are separated according to their size. Under native conditions, a protein with the same mass but different charge and shape would migrate differently in an applied electric field. The addition of SDS, along with a reducing agent, linearizes the protein by disrupting the tertiary structure, and the SDS coats the protein in a uniform charge per unit mass, which is approximately 1.4 grams of SDS per gram of protein. This negatively charged, linearized protein then migrates through the sieving matrix towards the positive electrode. Larger proteins are slowed down more by the sieving matrix, whereas smaller proteins are less hindered and can move more freely through the capillary. Capillaries offer many advantages over traditional gels. We can use very small sample volumes, typically 50 microliters. We can use 10 to 100 times higher electric fields without the negative effects of dual heating. We can apply voltages of up to 28 kilovolts, and for a CGE separation we generally use between 16 to 18 kilovolts. This increases the efficiency of the separation, leading to better resolution. We have on-capillary detection, and the instrument is completely automated, leading to greater walkaway time for the user. Here at the bottom we have a molecular weight ladder that was run on both a traditional slab gel and the HPCE 512. Traditional gels can take hours to run, stain, destain and analyze. Once the gel has finished running, the user then has to scan the bands to obtain quantitative information, which is time-consuming and laborious. There are also high levels of inaccuracy associated with trying to quantify bands on a gel. We can see that on the gel here that it has the characteristic U-shaped bands, which make quantification difficult. The HPCE, on the other hand, is fast. We can see that separation is achieved within 30 minutes. It's very easy to quantitate, as the area under each of the peaks gives concentration. It is highly repeatable, and we get excellent resolution. So here at the top of this slide we have a schematic representation of a conventional CE system. So we have a capillary, and each one of those bands within that capillary is a separated analyte. These move past the detector, and the detector measures each analyte only once. So this can lead to noise-related problems. Any lamp fluctuations, any instrument noise, or any analytes moving in the opposite direction can appear as peaks. Compare that to the schematic at the bottom, which is the HPCE 512. Here we have a photodiode array with 512 pixels, each one scanning at about 10 Hz. This results in hundreds of thousands of data points per analysis, which includes data on both time and band position. This generally results in at least a 10-fold improvement in signal-to-noise compared to a single pixel. This is because noise is dependent on the number of photons collected during the experiment. Splitting the detector up into 512 pixels means that we can keep resolution whilst increasing our detection area, hence increasing sensitivity. The instrument performs a self-check prior to each sample being run. This makes sure that the capillary is functioning well, that it isn't cracked or broken, and that it is adequately filled with buffer. We are also able to identify and discriminate against any species moving in the opposite direction to the direction of electrophoresis, or with an unexpected velocity. This is because the instrument generates a map of each analyte moving past each of the 512 pixels, and from this we are able not only to identify any species moving in the wrong direction, but also account for bubbles and lamp fluctuations. The excellent sensitivities achieved using the HPCE 512 enable the system to be completely label-free. This decreases sample preparation steps and offers a significant cost saving, as the majority of the expenditure for experiments can be due to the necessity for labels and dyes. This is the Rituximab intact antibody, so the antibody has not been reduced but has been treated with SDS. Here we have a comparison of two samples. The test sample is a sample that has been reconstituted and is ready for infusion, and the reference sample is the concentrate from which the test sample is made up. Both samples have been run at a final concentration of 0.5 mg per ml. The test sample is in green and the reference sample is in red. We can see that there is a decrease in peak area of the main peak, peak 7, in the test sample, as well as a decrease in percentage peak area. Changes in peak area show changes in concentration, whereas the migration time corresponds to molecular weight. Each of the labelled peaks can be mass assigned by means of a calibration curve. The decrease in peak area of peak 7 in the test sample could be as a result of degradation of the intact antibody. There is also an extra peak, peak 8, in the test sample, which may indicate aggregation. These subtle changes in profile could indicate alterations in drug potency and efficacy. One of the main advantages of this technique is the minimal sample preparation steps that are required. In this case, the samples were diluted into a buffer containing SDS and heated for 10 minutes, following which they were transferred to the instrument for running. Once the instrument is running, no further user intervention is required, allowing the user to be freed up for other tasks. The data is automatically collected and can be opened as soon as the sample has finished running. Here we have the Rituximab force-degraded sample. The 40-degree incubation was used to degrade the sample and show the effects of inappropriate storage. The reduced sample is shown in blue, and the non-reduced sample is shown in green. The non-degraded samples are shown in the inset for comparison. We can clearly see that a greater number of peaks are observed in the degraded samples, with a number of small, low molecular weight species being observed. This highlights the excellent sensitivity of the system, which is able to accurately detect and quantify small peaks, even in the presence of larger peaks. And again, each of the labelled peaks can be mass assigned by means of a calibration curve. The clear differences observed between the degraded and non-degraded samples show the usefulness of the HPCE 512 system in detecting and quantifying degradation in an antibody sample without the need for expensive labels or dyes. This is highly beneficial in determining whether samples have been appropriately stored and whether they are still in a good enough condition to administer to patients. The next mode of electrophoresis that was used was capillary zone electrophoresis, or CZE. CZE was used for charge variant analysis. Monoclonal antibody charge variants can occur as a result of many types of post-translational modifications. CZE was carried out in a simple buffer system. Unlike CGE, there is no SDS present in this system, and the proteins are allowed to remain in their native conformation, so there is no reduction of samples. Buffers such as borates, phosphates, and acetates are typically used for CZE, and there are many additives that can also be included in the buffers to alter the properties of the capillary surface. Buffers are typically chosen for their low conductivities, good buffering capacity in the pH range of interest, and low UV absorption. In this mode of electrophoresis, the charge of the protein plays a more important role than mass, and any charge variations caused by different post-translational modifications can be easily picked up. The differences in charge and isoelectric point cause the protein subspecies to migrate differently relative to one another when placed in an electric field, resulting in separation of these species. In this way, CZE is an orthogonal technique to CGE as it provides information that may not be possible to obtain from CGE alone. For all the CZE experiments, minimal sample preparation was required. The samples were diluted into sample buffer and then placed directly into the instrument for running. This quick sample preparation protocol results in significant time saving for the user. Here we have the data for the charge variant analysis of Infliximab. These samples were run at a final concentration of 0.2 mg per ml. We have a sample that has been appropriately stored between 2 to 8 degrees and one that has been degraded at 40 degrees. We see a significant decrease in overall peak area for the degraded sample, and if we look at the tables at the bottom, there is an overall shift from the more basic species to the acidic species following storage at 40 degrees. Acidic and basic charge species are formed from different chemical degradation pathways. For example, acidic species can be formed by deamidation, whereas basic species can be formed by the oxidation of methionine, cysteine, lysine, histidine, and tryptophan, or acylation. These peak areas we see here are automatically generated by the software, so parameters such as peak area, migration time, corrected area, and percentage area are outputted as soon as the peaks have been identified. This makes data analysis very easy. This CZE technique provides a high resolution of separation combined with fast separation time. This type of analysis would be highly beneficial to monitor production variations between monoclonal antibody batches. Changes in the charge profile may affect Fc receptor binding, potency, or pharmacokinetic properties. Data from charge variant analysis can be combined with other techniques measuring biological activity to assess the effect of manufacturing variations. Here we've carried out repeatability analysis on the charge variants of Infliximab. So this is the 0.2 mg per ml controlled sample, and 6 replicate injections were carried out, and the percentage relative standard deviation of peak 1 was calculated, and this was found to be 2.283 for time and 2.882 for area. The percentage relative standard deviation is a measure of the precision of a measurement and is the ratio of the standard deviation to the mean. The peak time is related to either molecular weight in CGE runs or to identification of protein species in CZE runs, and the peak area relates to concentration. The excellent repeatability obtained with the HPCE 512 makes it an ideal tool for analyses requiring accurate measurements and applications such as QA/QC and stability studies. The repeatability values seen here are significantly better than values obtained for many other analytical techniques. The final stage of assessing the suitability of an antibody drug product is evaluating its binding to the target molecule. So here we've looked at the binding of Infliximab to TNF-alpha using CZE. The label-free nature of the HPCE 512 enables the monitoring of these binding events without the interference of labels. Carrying out binding studies on the HPCE 512 is extremely easy. The two proteins being investigated are generally run separately, and this is then followed by a run once binding is expected to have occurred. It may also be possible to carry out the incubation step in the instrument's carousel, depending on the temperature required, and inject at different time points. We would then generally expect to see either a shift in migration time following binding, or changes in peak area which can be caused by alterations of the intrinsic absorbance of the proteins once binding has occurred. Here we have the data for the binding of Infliximab to TNF-alpha. The top trace is 1 mg/ml TNF-alpha, and the middle trace is 0.2 mg/ml Infliximab. These two solutions were then combined at room temperature for 5 hours with shaking. The bottom trace shows the results obtained following this incubation. We can see that the Infliximab peak has been lost, and there is a slight shift in the TNF peak. These results indicate that binding has occurred. The TNF-alpha in this study was present in excess, and we are in the process of carrying out experiments at various concentrations of Infliximab and TNF-alpha to confirm these findings. Other data has also shown that the time course of binding can be followed by running samples at different time points during the incubation step. We were able to see a gradual shift of the TNF peak as binding took place. That data isn't shown in this presentation. These preliminary data show that binding events can be followed using the HPCE 512, which can take the place of expensive ELISA measurements. The data presented here demonstrate the ability of the HPCE 512 capillary electrophoresis platform to completely characterise a monoclonal antibody product. We used capillary gel electrophoresis and capillary zone electrophoresis to obtain information on the structure and functionality of the antibody samples. We obtained excellent repeatability for peak time and peak area. The data shown here demonstrate the versatility of the HPCE 512 to carry out a range of analyses using a single analytical platform. This would lead to significant savings in capital expenditure, as a number of analytical tools can be replaced by a single system. The low running costs of the HPCE 512 also make it ideal to act as a triage for more expensive techniques. Thank you very much for your attention. I hope you found this webinar useful and that it has highlighted some areas in which this technology can be used in your labs. We'd be happy to discuss any thoughts on potential applications and answer any questions.
Well, thank you for that. I hope you found that to be of interest. As I mentioned before, we're going to have a question and answer session now, so if you've got any questions at all, please feel free to write them in the dialog box there to Deepika.
Thank you, John. We've had a question asking about why we don't need to use labels or dyes, and that's because of our multi-pixel detection, which greatly improves sensitivity of the system. So we have 512 pixels that are scanning at about 10 Hz. So for each sample that's injected, we get hundreds of thousands of bits of data. This data is amalgamated, and this improves signal-to-noise by at least 10-fold compared to a single pixel. And because of the increased sensitivity, we don't need to use labels and dyes. We've had another question coming through, and that question is, what are the minimum concentrations required for analysis? This greatly depends on the analyte of interest. Because we're looking at the intrinsic absorbance of the analyte, it just depends on how well it absorbs. So for an amino acid, we typically look at low micromolar range of concentration. So for aspartic acid, we get an LOD in the region of 0.2 micromolar. For proteins, this is slightly different. So for an IgG, we typically get a limit of detection of about 1.3 micrograms per mil. We've had another question through, which is, what is the cost per analysis? Because we don't use labels or dyes, our running costs are very low. We typically use very standard buffers that are available in most labs, borates, phosphates, for example, for CZE. So the only cost is really the vials and the capillary itself, which are very cheap.
If there's any other questions, please feel free to type them up there now. These might not be related directly to the presentation that you've just seen, perhaps they're related to how the technique can be used for your sample. So if you've got any questions, please feel free to just type them up and we'll wait for a few minutes whilst other questions come in. We've got a question here, whether or not it's possible to keep the sample temperature controlled whilst it's analysed.
Yes, absolutely. So this helps with the repeatability of our system. So we're able to control the temperature of both the capillary block itself and the carousels where the samples are located. So the capillary block is controlled by a Peltier system, and we're able to control temperature to within 0.1 of a degree. We're also able to control the temperature of the samples within the carousel, and this obviously greatly improves repeatability of the system. That, combined with our ability to exclude noise, gives us the better repeatability compared to other CE systems. We've had another question about what the minimum volume is required for analysis. We typically require 50 microliters per sample, but this sample can be injected a number of times because the instrument only takes up a few nanoliters per injection. So to carry out all of the analysis that we looked at during the presentation, we'd probably only need between 1 to 200 microliters of sample.
Okay, well, if you have any further questions, feel free to get in touch with us. Like I say, it might be related to the presentation, it might be related to your own samples. So whether you're watching this live or you're watching the on-demand version, feel free to contact me. My name's John Cooper, and my email address is john.cooper, that's john.cooper@horiba.com. You can ask your questions at any time, and we'll try our best to get back to you as soon as we can. Thank you very much for sparing the time to watch us this afternoon.